Well, hello everyone. Thanks so much for coming. So today we have Dr. Guadalupe Peralta, and I have been following her work for a very long time because it's broadly about species interactions, mutualistic networks, I would say very importantly, but amazingly, you've also worked in all of these different systems, predator-prey, herbivory, and I feel like whenever my Google Scholar alert goes off with like, you might be interested in this, it's a paper by Guadalupe. So we had the privilege of meeting in Argentina, but where she's from, but she did her postdoc. She did her graduate work in Argentina, then she did her postdoc in New Zealand. And now she's back in Argentina to start a professorship with CONICET, pronounced correctly.
Yep.
Which is a really incredible positions, which are fully research. And yeah, so with that, I'm really excited to hear about your work.
Oh, thank you very much, Lauren, for the introduction. Thank you, everyone who's there, and I hope you're all coping with the current situation. And as Lauren said, I'm going to talk about species interactions and what happened at the backstage of species interactions. So back in the days before the digital era, movies used to be made by pasting a set of photographs. And sometimes I have this idea that in ecology, we tend to do something similar. We take samples from different places at different locations, different times, and then we combine them to tell a story. And a lot of the interactions that you can see in these pictures occur or last only for a few seconds, but they have a huge impact on our lives. According to the Food Agriculture Organization, approximately 75% of the world's crops that produce fruits and seeds for our consumption depend, at least partially, on pollinators. So this is a huge amount of food that we consume, and the type of food that we should be consuming even more, right? The healthy type of food. Another example of how important species interactions are for our lives is the typical example of biological control. And as you may know, for multiple agricultural commodities, biological control provides benefits of billions of dollars per year. This represents a huge impact on our economies, and like it or not, economies impact our lives a lot, right? So these two examples that I'm showing you here, the flower with the pollinator and the host with the parasitoid, the pest with the biological control agent, are only interactions among two species. But of course, these interactions don't occur like this in isolation, but species interact in very complex ways, right? So in this community, hypothetical community I'm showing you, there are lots of interactions between lots of different species. And this web of interactions is what we call a species interaction network. And here in this example, I'm showing you interactions between plants, which are the green circles, and animals from a different trophic level, which are the yellow circles, right? And of course, the links that link the circles represent the species, the interactions that occur among these species. And within communities, there are different types of interactions. There are mutualistic interactions like the one I was showing you before, talking to you about pollination. So here, as you can see, plants and pollinators interacting. There are other types of interactions like frugivorous interactions. So the bird eats the fruit and hopefully disperses the seed. And there are other types of interactions like antagonistic interactions among, in this case, herbivorous caterpillars and plants. And of course, species interact in different ways and with different frequencies. And here, with the width of the links among species represent how frequently two species interact. So my research is based on these interaction patterns at the community level. And what I intend to do with my research is to try to find out what do these patterns of interactions tells us and how they can be useful for us. So this is what my seminar is going to be about, and it's going to be divided in three sections. The first one is going to be about how species interaction patterns can help us understand ecosystem functions. The second one, it's going to be about the drivers, what are the mechanisms that drive species interactions. And finally, I'm going to talk about what are the applied implications of the results I'm going to show you before. So let's start with species interaction patterns and how they can help us understand existing functions. So as I was showing you before, species interact in really complex ways in nature. And of course, to sample all these different types of interactions at the same time would be really difficult. It would be logistically impossible, probably. So what we tend to do when we study species interactions is to focus on specific groups of species. For example, we tend to study plant pollinator interactions, or we tend to study plant herbivore interactions, or predator-prey interactions, like this example of caterpillars, hosts, and parasitoids. They're predators, right? And it has been shown that the pattern in which these species interact in communities can help us to understand what are the mechanisms that drive ecosystem functions. And let me give you an example. So in this community, here we have two communities, A and B. Both communities have exactly the same species composition, same species abundances, but as you can see, there are different links among the species. So they have a different interaction network structure, right? Species interact differently on each community. In community A, as you can see, most of the parasitoids do not share the resources, right? So they split the resources. They have a high resource use complementarity. On the contrary, in community B, parasitoid species do share, all the parasitoid species share at least one host species with another parasitoid species, right? So we can say that resource use complementarity in this community B is lower. Now, you may be aware, you know that the biodiversity ecosystem functioning relationship, it's been really studied, and one of the mechanisms that it's supposed to drive this relationship, higher biodiversity, higher ecosystem functioning, is this one of resource partitioning, right? Niche partitioning. So one of the things that I decided to do for my PhD was to test this, was to test whether resource use complementarity is a driver of attack rates of parasitoids. So for my PhD, I collected, I sampled interactions among caterpillars and parasitoids in native and plantation forests in New Zealand. So we basically collected all the caterpillars, reared them, and found out which parasitoids came out of them. We collected thousands of them, so we had a huge data set. And with this data set, well, we knew the interactions, of course, because we observed them, so we could measure how much complementarity was there among parasitoid species from one community. And of course, we could also estimate the parasitism rate of each caterpillar species and at the community level. So what we found was that indeed, the higher the resource use complementarity in a community, the higher the parasitism rate of those communities. So each circle in this figure represents a community. And this pattern of complementarity in resource use and functioning has been also observed for other systems. So more recently, last year, this study came out that shows exactly the same, but for plant pollinator systems. So basically, McGrath et al. Showed that higher pollinator complementarity increased plant reproductive success, so the number of seeds produced per fruit. And similarly, a couple of years ago, Daniel Garcia's group found out exactly the same pattern for plant frugivorous interactions. So basically, they show that when frugivorous birds split their resources, they use complementarity increases, and it also increases the seed dispersal function of those communities. So this is really interesting because it's a pattern that it repeats in different study systems. Another way of thinking about this pattern of interactions is redundancy, right? So in community A, there is not much redundancy in in how many parasitoids attack each host, right? So basically, each host is, or almost all hosts on community A are attacked only by one parasitoid species, right? If something happened to one of those parasitoid species, then the host is released from its predators, right? So we can say that community A, has a low redundancy, whereas community B has high redundancy because a lot of parasitoid species attack the same host species. And this is one of the mechanisms that could influence how stable parasitism rates are across a space or across time. And this is something that I also tested during my PhD with the same data set I was telling you before. What I evaluated was whether the spatial stability of parasitism rates was related to redundancy in the attack of hosts. And what we found was that indeed there was a relationship. So in the x-axis we have redundancy, and in the y-axis we have the spatial variability of parasitism rates. In this case, a spatial variability, it's kind of like an inverse measurement of stability. So higher variability means lower stability. So basically what this figure tells us is that when redundancy increases, spatial variability of parasitism rates decreases, which means they are more stable across a space. They are more constant across a space, those parasitism rates. So as a first conclusion, we can say that species interaction patterns at the community level can help us understand ecosystem functions and the changes and the stability of those functions. So this means that understanding how species interact, when and where species interactions occur, could be really useful, right? Could be really useful to inform us about these ecosystem functions that have such a huge impact on our lives. But of course, to find out the how, when, and where of species interactions, instead of thinking or looking at the movie, we need to look at what's happening behind the scenes, right? We need to understand what's making Superman fly in order to make him fly again in another movie, or at least predict when it's going to fly, right? So this brings me to the second section of the talk, which is what are the drivers of species interactions? So, as you know, species interactions not only depend on the co-occurrence of species, right? But species interactions depend on a lot of different factors. For example, some of which I wrote here, for example, morphological traits of species, species behavior, their physiology, species abundances, their phenologies, and of course, the environmental conditions where the species are co-occurring, right? When we try to predict interactions and when we try to evaluate what are the influence of these factors for the occurrence of interactions, we assume that all these factors are equally important for the occurrence of all the interactions across the community. But this could not be the case. And let me give you an example. In this community, we have plant and bird interactions. So the birds eat the fruits of the plant, and in this type of of interactions, we know that morphology is really important. So the bill size or big size of the birds, for example, needs to match with the fruit size of the fruits the birds are consuming. So there needs to be trait matching among these species. And not only bill size and fruit size, but also the height at which the fruits are offered by the trees or plants need to match also the height at which birds fly, for example, right? So we know that morphology is important, and we assume that it's equally important for all the interactions among all the species, among all the birds and all the plants, right? But species have different characteristics that could make this trait matching more or less important in the occurrence of interactions. For instance, here we have a generalist bird, right? The blackbird is a generalist species, so it fits on a lot of different plant species. And we know from previous research that generalists tend to have trait values that are more similar to the average trait value of the community, right? Whereas the most specialist species tend to have the most extreme trait values of the communities, right? So there are also other characteristics, like, for example, the biogeographic range of the species, whether the species is native or exotic, which could also influence how important traits or trait matching is for the occurrence of interactions. So we know that interactions that occur among native species have had a really long time to co-evolve and to co-evolve traits. So it could be possible that interactions that occur among specialist species and those that are native particularly have a higher trait matching compared to other interactions more generalist species or exotic species, right? So we decided to try to answer this question. Is trait matching equally important for all interactions among all the species, or does it depend on the characteristics of the species? And in order to answer this question, we use this large data set that George Perry, our collaborator, collected in New Zealand. So he basically collected all the interactions that occur among plants and birds that have been published in the literature from New Zealand. And this is really interesting because there are a lot of native species in New Zealand, of course, but there are also a lot of exotic species. And we also collected data on the traits, the species traits, from museum specimens, from also the literature, from herbarium specimens. So we created this huge database of species interactions among birds and plants and their traits, right? So with this database, what we estimated the generalism degree of the species. So basically with how many partners does that species have. And we also estimated what was the trait matching degree between the species that are interacting, right? And what we found was that when species are more generalist, so when the interacting species interact with a lot of other species as well, the trait matching of those species is lower compared to species that are more specialists. So trait matching is more important for interactions that occur among specialist species. But we also have that information about the biogeographic range or status of the species, right? So we knew which species are native from New Zealand and which are exotic. So we decided to assess whether this relationship was the same or whether it changed the slope of the relationship, depending on whether the interacting species were both native, both exotic, which could be exotic from different parts of the world, not necessarily from the same part of the world, or interactions among native and exotic species, right? And what we found is that trait matching is more important for interactions that occur among native species. So as you can see, the green line represents interactions between native birds and native plants, and then we have the yellow line that represents interactions between native plants and exotic birds, or exotic plants and native birds. And finally, the lower line, the purple one, represents interactions among exotic species. So we can see that in all the cases, the more generalist, the interactions among the most generalist species have lower trait matching. But overall, interactions that occur among native species have a higher trait matching. But then we started thinking, and we thought, well, would this pattern be repeated for other interaction types? So this is just one example of plant frugivorous interactions, but what would it happen with, for example, plant pollinator interactions? So we decided to test this question again using another data set. So this data set was collected by Diego Vázquez, Natasha Jacob, and Silvia Lomascolo, among other collaborators. So you can see Diego, Nacho, and Silvia in the pictures. They collected this plant pollinator data set in the Monte Desert in Mendoza, Argentina, so right next to the Andes. And basically, this data set contains information on plant pollinator interactions, also on the traits, on pollinator traits and flower traits. And we always choose, of course, traits that are related to the occurrence of the interactions, right? So, for example, proboscis length and corolla tube length, and body size, and the opening width of the corolla tube, right? So this type of traits. So again, we could, with this data set, estimate what was the trait matching degree of plant pollinator interactions. And we could also, because we knew the pattern of interactions among all the species that occur in this community, or most of the species at least, we could estimate what was the generalism degree of the interacting species, right? So same as before, in the x-axis, we have what's the mean generalism degree of both interacting species, and in the y-axis, we have the trait matching, how well the trait of the interacting species match. And what we found was that, same as before, the more generalist the species are, the lower the trait matching of those species, right? So again, for this plant pollinator system, we have the same conclusion. Trait matching is more important for interactions that occur among generalist species. Unfortunately, for this data set, we didn't have information on the biogeographic status of the species, so whether they were native or whether they were exotic species, right? Except for the obvious the obvious ones like the honeybee, right? So we couldn't test the second hypothesis, but at least for the first one, we can observe exactly the same pattern as we observed for the other data set of plant and bird interactions. So for all the interactions in the community, this is basically the same, just the same results as I showed you before. Specialization is basically the same as generalism, just a different name. So we can see here that a specialization influences the trait matching degree of the species that are interacting. So it affects how morphology influences the occurrence of an interaction. So we decided to go a step forward with this data set and with these questions that we were answering, and we decided to assess what would happen with interaction frequency and with the temporal stability of those interactions. So does trait matching also affect the frequency with which species interact and how stable they are across time? So luckily, this dataset has also, this plant pollinator dataset collected in Mendoza also has information on interaction frequency and on temporal measurements of interaction frequency. So we could estimate both well, interaction frequency and the temporal stability of those interactions. But to assess what are the effects of something on interaction frequency, we have to include the abundance of species, right? Because when a species is more abundant, it could happen that it randomly encounters more individuals from other species. So so the chances of having a higher interaction frequency are higher, right? So in order to assess the effects of morphology, we need to include abundance or kind of remove the effect of abundance to truly assess the effects of morphology. So luckily we had information on species abundance for that data set, so we included that variable as well in our model. And of course, abundance could have a direct effect on both interaction frequency and the temporal stability of this interaction frequency, or it could indirectly affect interaction frequency by influencing species phenological overlap, right? So here, phenology with the clock, what I'm representing is how well the two interacting species phenologies overlap, right? We know from previous research that the species that are more abundant tend to have longer phenologies, so there is a higher chance that their phenologies are going to overlap with the phenologies of other species. So we estimated what was the phenological overlap of the interacting species and also included it in our model as a predictor variable of interaction frequency and the temporal stability of interactions. And this is what we found. The black arrows represent positive effects. So as we found for the occurrence of interactions, the higher the specialization of the species, the higher the trait matching of the species, and the higher the interaction frequency and the temporal stability of those interactions, right? So those results remain with the same sign. But probably more or as exciting as this result was that the effect of abundance was on interaction frequency and the temporal stability of interactions was most of it was indirectly via phenological overlap of the species, right? So abundance influences species phenologies, which in turn is going to influence the interaction frequency of species, increase interaction frequency, and increase temporal stability of a species. This is quite interesting because when we use models to try to predict interactions, for example, we tend to include the species abundances, but a lot of the times we leave the species phenologies aside. But if we include the species phenologies and species phenological overlap, we are going to be incorporating a lot of the effects of a species abundances in our models, right? And sometimes estimating species phenologies or phenological overlap of species, it's easier than estimating species abundances. And finally, we thought, well, what are the effects of these predictor variables on the final existing function? So pollination, right? Which is what a lot of the times we care to conserve. And we found that both phenological overlap, so species phenologies, and species morphologies, so trait matching, indirectly influence pollination success by increasing interaction frequency, right? So this is really interesting, and this is basically takes me to the second conclusion that species interaction drivers are not equally important for all interactions. Then they depend on species characteristics, and these drivers can help us understand not only species interaction dynamics, but also what are the changes in ecosystem functions that we are observing. And finally, in for this last section, I saved this topic, which is one of the questions that I would say most of the ecologists that do more basic research are terrified to hear. What are the applications of your research? Why is it useful? You know. So, anyways, I'm going to talk about exactly that. What are the applied implications of the results I showed you before? And for this section, I'm going to focus on biological control and the non-target impacts of biological control. So, as you know, biological control is a tool that we use when we have a focal host in a community, which abundance is causing damage in a plant species, for example, that we care about. So biological control looks for a biocontrol agent. In this case, it's a parasitoid, and the host is caterpillar. And what it does, it's basically to look for a biocontrol agent that we know it's going to attack our focal host and it's going to decrease its abundance. So it's going to remove the problem, right? So when we introduce a biocontrol agent, it could happen that this biocontrol agent interacts with other species from the community, like these other hosts. In general, we don't want this because this can cause other effects. So, like a decrease in the abundance of the second host species, for example, and we don't really want to change other species abundances in the community, right? So that's why we tend to choose to avoid these direct impacts on other host species. We tend to choose the most specialist biocontrol agents that only attack our focal host, right? However, even when we find our biocontrol agent that only attacks our focal host, changes in the abundance of a host can affect other species, like other parasitoid species in this case, which in turn can cascade and affect other host species in the community. And in this case, for example, release other host species from parasitoids control, right? So these are called indirect effects, in this case, via shared natural enemies, right? This is not only a thing. Indirect effects, it's not something only from biological control, right? When we are using pesticides or when using any method of control, of pest control, we should be thinking about these indirect effects. And we should be really thinking about these indirect effects because they have a huge impact on the community, on both existing functions and also species abundances. So it has been shown that indirect effects via shared natural enemies determine up to 31% of the variation in herbivore abundances. So this is a huge proportion of the variation in species abundances. So indirect effects are playing a huge role in what happens in communities. So in order to assess which are the direct and indirect impacts of, in this case, biocontrol agents, if we know the pattern of interactions, so how species interact among each other in a community, we can actually predict what are these changes, these impacts going to be, right? So again, studying or working with species interaction networks can provide us information with which we can decide which is going to be the best biocontrol agent we can use. However, knowing all the interactions that occur in a community, it's not an easy task, right? That requires a lot of effort. That requires a lot of sampling effort and a lot of money that sometimes, well, we don't have, right, to do the work. And that's why predicting interactions, the prediction of interactions, is really important. And this is something that also network ecology can contribute to. Of course, predicting interactions, it's not an easy task because as I was mentioning to you before, interactions, the occurrence of interactions and the frequency of interactions depend on a lot of different factors, right? However, there are people working towards it, and I'm going to give you an example. So, as I just mentioned, the prediction of interactions requires a lot of knowledge that sometimes we don't have. So researchers that are working towards improving the prediction of interactions tend to focus on these three factors. So researchers use species traits, species abundances, and chronologies to try to predict interactions. And I'm not saying that these are the most important factors, but one of the reasons why they are important, of course, but how easy they are to measure, it also plays a huge role in why they are used, right? It's way easier to measure traits of species than to measure physiological traits of species, right? Or behavioral traits of the species. So anyways, using these three factors that predict, that contribute to the occurrence and frequency of interactions, one of a master's student in our lab group, Hannah Kotula, you can see her in the picture, she used these factors, these traits, and combined with machine learning approaches to try to predict what would be the direct and indirect impacts of biocontrol agents. So basically what Hannah did, she luckily she had two big data sets available, host parasitoid data sets. So these data sets contain both species interactions at the community level. They were quite complete, and also they contain traits of the species, right? So she used different models. I'm just going to show you the results from one of them, the random forest model. And she used this model, random forest model, with one of the data sets she had available. So basically, she combined these two components. And what the model does is that it learns from the data which host parasitoid trait combinations are likely to correspond to an interaction. So the model looks into these trait matching rules and how they change according to their specialization degree, and generate a set of rules. And then, based on those rules, it applied those rules to new species. So basically, this is where the second data set comes into play. So it applies those rules to the new species of the second data set and predicts what would be the interaction probabilities of these new species, right? So what Hannah thought was, well, if these interaction probabilities that the model generates correspond with higher interaction frequencies observed in the second data set, then we could use these type of models, for example, in biocontrol risk assessments. We could use them to say, well, this biocontrol agent is going to potentially interact with all these species, and we could estimate what would be their non-target impacts. So this is one of Hannah's results. As you can see, in the x-axis, we have the model predicted probability. So this is the probability of two species interacting according to the model. And in the y-axis, we have the observed interaction frequency. So the actual frequency with which those species interact in the empirical data set, right? The different symbols represent different ways with which Hannah trained the model, different subsets of the dataset. I'm not going to go into those details, but the trends are all the same. So for the general idea, it doesn't matter. So as you can see, what Hannah found was that the higher the predicted probability, so when the model predicts a higher probability of interactions among a host and a parasitoid species, that corresponds with a higher interaction frequency observed in the field. So This means that this type of models, the random forest models, could be used in risk assessment to predict attack rates and non-target attack of host species. So as a final conclusion from this last part is that the prediction of interactions can help us assess non-target impacts of biocontrol agents, and not only biocontrol agents, but also exotic species. For example, when a species is introduced, we could assess what are going to be, or at least partially, the impacts of that species in a new community. So as a summary, so the first part of this seminar was I show you how species interaction patterns can provide useful information on ecosystem functions and what are the mechanisms that drive ecosystem functions, their stability, and I believe that it's really important to understand why things are changing in order to be able to predict why or when they are going to change again. And in order to be able also to truly manage ecosystems and to develop proper management and conservation strategies, right? We need to be targeting the cause, not the effect. So on the second part of this talk, I show you how species characteristics dictate the level of importance of the different factors that determine species interactions. So the drivers of the species interactions don't have the same importance for all the interactions, right? The different characteristics of a species are going to influence the degree of importance of these factors. So again, we need to keep going step backwards and see what's actually affecting, what's the cause or why things are changing in a certain way. And finally, with all this knowledge that species interaction networks and species traits and functional ecology provides us, we can improve the prediction of interactions, and we can really use that to assess non-target impacts of introduced species. As I show you the example of biocontrol agents, but as I also mentioned to you, the effects or impacts of exotic species. So finally, I want to thank you very much for the invitation to participate in this seminar. And of course, I want to thank the collaborators. This work wouldn't have been possible without them. And so thanks a lot. And I want to mention them from the top. The guy from the hat is Matthias Delling. And then towards the right, we have Natasha Chakov, Rafael Didam, Silvia Lomascolo, Tatiana Rand, Carol Frost, Diego Vasquez, George Perry, Hannah Kotula, and Jason Tilianakis. They form my network of interactions. And of course, there is a lot of other people contributing and collaborating behind the scenes. So thank you very much. And I'm happy to answer any questions you may have.
Thank you so much, Lupe. If you guys want to ask questions, oh, looks like questions are already happening in the chat. So how common is the use of biological control, parasitoid wasps, in agriculture compared to the use of other means like pesticides and stuff? And we just start there. I guess that's, yeah.
Well, I don't have numbers to answer that question, but I would say that in the scale of things, it's very small compared to pesticides and herbicides. And yes, I don't know how is it over there, but here you go out and you usually see these huge fields of monoculture and they don't use biological control. So they just hire planes or hire people and just spray. So there is a huge area of the earth that's covered by these monocultures, and most of them, I would say, use other methods, not biological control.
Yeah, it's the same here. And pretty similar. Monica also asked, are there any shifting plant phenologies due to climate change in your study sites? And how fast are the herbivores and parasitoids adapting to it?
In our study sites, we haven't seen it, but there are studies that look into how climate change, yes, is influencing species phenologies. And the things that you need to have really long time series studies in order to be able to evaluate that. And that's really hard to get, as you know, because you need to have funding for a lot of years, which is not very common to have. But yes, there are a couple of studies that have looked into that, and they can see already some changes in species phenologies according or depending on influenced by climate change. So yes, I think the more data we collect and more the more time steps we are able to keep sampling, or the longer the time series we can collect, the more the effects we are going to be able to observe.
Michelle asks, do you ever see species change its status from specialist to generalist or vice versa? It's like that classification on the x-axis.
Well,
yes,
there's more there when it becomes an invasive or we know it's, you said a lot about what happens when they're in their native habitat, and you also talked about invasives, and I'm just curious if it is a specialist in its native, is it always a specialist when it invades or is introduced, or does it change?
Yes, well, at least from the data sets I have in mind, usually the species that are that tend to invade or that tend to colonize or adapt well to new habitat tend to be generalist species. So those are the ones that I guess because of their generalism degree is one of the traits probably that make them, or characteristics that make them more flexible to adapt to a new place. Mm-hmm. So when we estimate the degree of generalism of a species, we use all the data we have available for that species, right? So if you measure species generalism at just one time step, you might observe, yes, that species, here it's just feeding on one host, and then on another month, it's feeding on two different hosts. But when we estimate species generalism, we combine all the data we have across all samples.
Thank you.
That's all the questions in the chat so far. Anyone have any thoughts or last questions? So I have a question about that, Lupe. So kind of going off the generalism specialism question. So I often feel like there's a kind of a chicken and the egg situation of the trait matching and the specialization, right? Where if you have really strange traits that you can't interact with a lot of different things, you tend to look more specialized. Like which comes first, or was there a way that you guys thought of that? Maybe it was in taking all the different interactions from across different communities, but something that has always hurt my brain a little bit.
Yes, we thought about that, and I agree with you. You can see it from both sides. I don't know what would be the. I don't know whether there is a right answer. I mean, or whether we can answer that with the current data we have. But Yes, there is a bit of a circularity. So both, I guess that arrow could go both ways. And yes, maybe if you include species like the evolutionary history of species, and you try to assess whether those traits have co-evolved. Well, but again, and yes, you would need to have information on whether the species was before also a specialist, or yes, I don't think we have the data, or I don't know actually of data available that would allow us to solve that question, that arrow, the correct direction of that arrow. But yeah, it's a really interesting idea to keep thinking about it and try to test.
Great. Well, we're gonna have a tea time. So I guess, Peter, do we take a little bit of a break and then reconvene in a bit?
Yeah, you need time to make tea.
Okay, well, thank you so much, Lupe, and thank you for coming.
Thank you very much.